Okay, I'm based just down the road. I'm a little bit of a fish out of water here because I'm myself a molecular biologist and a professor of biology at the University of Cambridge. But what my lab, about 25 people just down the road in the Gurd Institute, we've realised over the last few years that what we've been doing in cancer is very, very relevant for genetic diseases, particularly rare genetic diseases. So I just want to start out by saying we are in the midst of a revolution in the life sciences of medicine. So there are many things going on, but of course we know through next generation sequencing and other approaches, we're getting insights into a whole range of rare genetic diseases. We know the genetics of many diseases now, be they rare diseases or more sporadic ones. And through advances in cell and molecular biology, we can actually generate cellular models and animal models that in ways that we couldn't even believe were possible a few years ago. So I think we're in a fantastic position now to translate this insight into the genetics of disease, into understanding the fundamentals of disease, and out of that, I believe, we can identify new opportunities. So we know gene defects cause many diseases, rare diseases, but also cancer. But it turns out that genes don't work in isolation. The products of genes actually work with other products of other genes. And it's been well known in model organisms, but we also know in humans for many years, that there are genetic interactions. So here, for example, one end of the extreme is something called synthetic lethality. Here, the defect in gene A doesn't cause death, doesn't cause disease. Gene B defects doesn't cause disease, but when you combine them both, you actually get a dead cell or a disease situation. At the other end of the extreme, you've got what we call synthetic viability. Here, a defect in gene A gives a disease state or a cellular abnormality. And we find out, and this is well extrapolated and very well established in model organisms that we now know in humans, that there are certain genes that if you knock out or make mutations in, you actually rebalance the disease phenotype. Okay. So these genetic concepts have been a major focus of my lab for the last few years, to understand biology, but also translate that into medicine. To very briefly summarise, I set out about 19 years ago to try and extrapolate the synthetic lethality opportunity in cancer. And the idea here is if a cancer cell has a certain gene defect, like a BRCA1 mutation, we may be able to identify another gene and another gene product that that cancer cell is very reliant on. But long story short, a company called Kudos that I set up has developed the first in the world synthetic lethal medicine. The company was acquired by AstraZeneca, but basically what we're now seeing with this registered drug is it's having major effects in patients with BRCA1 and BRCA2 mutations with very little side effects. This is extending their lives, and AstraZeneca are going to make billions out of this. I made a bit, I'm not going to make any royalties, but I set up the company that led to this new medicine. So this new medicine, which is based on genetic defects, one end of the spectrum, if you like, is basically the first class drug, first synthetic lethality medicine in the world, and it's also the first cancer drug launched with a genetic diagnostic as part of its basis. So my lab's still interested in that. We're funded by Cancer Research UK, but we're also now interested in exploiting the other side of that equation brought out at the beginning. So if we have this situation here, if this gene B makes a druggable target, then if we can develop drugs against gene B, we'll be able to alleviate the symptoms caused by a gene A defect. And now, over 3/4 of my lab, one way or the other, that's about 20 people now, is focused on trying to extrapolate and understand that and connect that to human disease. We're doing this in various areas, but just one example I want to highlight. We published in the journal Science just a couple of years ago how we focused on a rare genetic disease called Hutchinson-Gilford progeria, very rare genetic disease. And basically, we did a cellular screen. We learned, or Delphine Larry in my lab learned, that when look at cells from these patients, or you genetically engineer cells, our normal cells in culture, to have this mutation, they look funny under the microscope. And basically, we screen for drugs that rebalance that phenotype, that make them look normal. And then through using chemical genetics, we identified the drug target, and we've now taken compounds into animal models. So this is an animal model that we're doing in collaboration with our colleagues in the Sanger Institute, where the mouse has exactly the same mutation that the patient does. And the mutant mouse has a phenotype, which is a progeria phenotype, so premature aging. And what we found is that with this compound, we're also knocking down the gene product, NAT10, where to extend the lifespan of these mice. Now, we don't know whether or not this is going to work in human patients, but we're very excited about it. My postdoc Delphine Lario came back from the US just last week from a conference focused on HGPS, and there's a huge amount of excitement about this molecule that we identified through cellular screens. It's not screening animals, it's not screening humans, it's screening a cell for a phenotype, a property that you want to rebalance back to normality. So the majority of my lab is doing chemical screens. We're also doing CRISPR-Cas9 mutagenesis, random mutagenesis, the same types of methods that have been used in Drosophila and model organisms for many years, to basically try and find ways of rebalancing disease at the cellular level. And our view is that if you can understand the cellular basis of the disease and you can find a way of rebalancing it, if that is through a mechanism that's targetable by drugs, either existing drugs that could be repurposed or new drugs, you have an opportunity for treating that disease. So where I am right now is with this idea. 10 seconds. We know a lot about disease. We know about genetic factors affecting disease, increasingly now through rare diseases. And we know a lot about cell and molecular biology. It should be straightforward to get here, but there's this valley of death. And what I'm now talking to major funding bodies, particularly the Wellcome Trust, is to establish an institute, an initiative to start with, called the Darwin Initiative for Disease Biology, that I hope will grow into an institute, which is about basically, which is about bridging this chasm between these two things in terms of developing models in a systematic way. So my vision is that within 5 years we may have a new institute based in around Cambridge of several hundred people. That's my goal. The Wellcome Trust have expressed a willingness to seed this. And what I'm now trying to do is convince them, other funding bodies, and philanthropists to actually get into this. Because my view is that rare diseases, there are many, many rare diseases, but in the end many of these boil down to cell biology, and many of the key principles, mechanisms, lab experiments are similar between diseases, even diseases that are very, very different. So what I want to establish is the Darwin Initiative, or the Darwin Institute for Disease Biology. And I think this could be a place where in the whole world people would want to interact with in terms of exploring, understanding genetic disease, but identifying disease mechanisms. And principally, the key goal for DIDB will be to identify new therapeutic routes that we would then pursue. This is an academic venture. We pursue in-house, on our own, but also through partnerships with patient organisations and also charities. I'll leave it there. Thanks.